Hello, my dear students. Welcome to my today's online class of recombinant DNA technology. Continuation of vectors. Vectors part 2. I am Dr. Meera BK, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bengaluru 1. You can see my cell phone number and so also my mail ID on the slide. After the class, if any queries, do contact me through these venues. Now let us start our class on vectors. In my previous class, I had taught you about vectors, types of vectors, and I had discussed in detail about plasmid vectors. Now, in this class we are going to learn two more types of vectors, one bacteriophage vectors and the other one plasmids. In bacteriophage vectors, we are going to learn about lambda-phages as vectors. Okay? By now you must be knowing that bacteriophage is a virus whose host cell is bacteria, right? So we are going to learn about phages which have been exploited as vectors. One such thing is lambda-phage vectors. Okay? Now, what are phages? Phages are the viruses that replicate within the bacteria. The advantage of these phage vectors compared to the plasmid vectors is that they can take up larger DNA fragments. They can take up larger DNA fragments when compared to plasmids. So that if the size of your DNA insert is bigger, then you can always go for phage vectors than plasmid vectors. Okay? Now, you have to do some background exercise for this. Open your microbiology textbooks. Make a note on biology of lambda-phage. Here I am just going to brief you. Lambda-phage is a virus of E. coli, that is Escherichia coli, and it has been most thoroughly studied. Okay? Since we know the biology very well, people have attempted to develop that into a vector. Its biology goes like this. It has a molecular weight of 31 into 10 to the power of 6 daltons, and the genome has 4805 base pairs. So the length goes as 4805 base pairs. Now, this lambda-phage DNA molecule size can be increased only by 5% of the new DNA. Like if you want to retain the entire genome of lambda-phage as such, plus you want to add DNA of your insert, it should not exceed by 5%. That means it comes to around 3 kb. Because if the total size is more than 52 kb, as such now the size is 48 kb, if the total size becomes more than 52 kb, the packaging of the viral particle into the head becomes a problem. Now you have to learn about biology of lambda-phage vector as homework. Okay. So 48 plus 3, 51 or 52 kb. Conveniently, it can be packaged into lambda head structure. So if you are not removing any genomic part of lambda-phage, there is scope to add only 5% of extra DNA, that comes to 3 kb of new DNA. Okay. Now, the idea of developing beta vectors with lambda-phage came in the way with the discovery that lambda segments could be deleted, lambda DNA segments could be deleted without impairing viability. What I meant is, out of that 4800 and odd kb, if you remove part of it, then also lambda phage will be normal only. Okay. So people came up with the idea that by deleting non-essential regions of the genome, you will have more scope to add the bigger DNA insert. This made people to work on how to develop lambda phages into vectors. Okay. Now, removal of all or part of this non-essential region. You know, the non-essential region lies between the positions 20 and 35 of the genetic map. Okay. So, if you remove part or total of this region of the genome between 20 and 35, this decreases the size of the molecule by 15 kb. So, here you will get around 15 kb, and as such you can add 3 kb. That means as much as 18 kb of new DNA can be added, okay, without hampering the package process of lambda phage heads. Okay. And the lambda genome is so large that it has more than one recognition sequence for almost every restriction endonuclease. Okay. So this made people to exploit lambda phages as vectors. Okay. Now, they have designed so far two types of lambda phage vectors. One is insertion vector and the other one is replacement vector. We shall study both. Now coming to insertion vectors. Here vectors have a single target site for the insertion of foreign DNA. Okay, for the insertion of foreign DNA, and wherever there is a single target site, just a cut is made and in that you will insert the DNA. Such vectors are known as insertion vectors. What you do is, a large segment of this non-essential region is deleted and the two arms are ligated. When you do so, the genome size decreases by 20%. Okay. So such phages will have 20% less than the wild phage DNA. And they possess at least one unique restriction site into which you can insert DNA of your choice. The size of the inserted DNA fragment that an individual vector carry depends upon the extension to which non-essential region has been deleted. Okay, non-essential region has been deleted. Now you can see in the diagram, the first one is the insertion vector. You can see the linear molecule of lambda phage. On either side, it is flanked by causocytes or cohesive termini, what we call. And in insertion vectors diagram, there is one place for EcoR1, that is the place where there is target sequence for EcoR1. What is done is, this linear DNA molecule is given to EcoR1, a cut is made, and the DNA of your insert, DNA of your choice is inserted in this region. Okay. How exactly it is done, the diagrammatic representation I have given in the notes, you can just refer. Okay. Now, best example for this type of insertion vector is lambda GT10. I will come to this slide a little later again. Best example for insertion vector is lambda GT10. This can carry up to 8 kb of new DNA, which is inserted into unique EcoR1 site, and this site is located in C1G at the position 857. So insertional inactivation of this gene means recombinant molecules can be distinguished by clear plaques. So what is done in lambda GT? You will take the linear DNA molecule, you will identify the non-essential region. In that C1 region at the position 857, which contains the target site for eco-R1, there a cut is made, digestion is brought about with the restriction enzyme eco-R1, and then in that place you will insert the DNA of your choice. Up to 8 kb of new DNA insert can be put in insertion vectors. This is what I wanted to tell you about insertion vectors. Coming back to the slide, this is the place where the place of eco-R1 where the cut can be made and your DNA can be inserted. Then people have designed another type of vectors making use of lambda phage only. They are called replacement vectors. You can make it in the diagram. The replacement vectors have two recognition sites. They will have two recognition sites instead of one as you see in insertion vectors. So you will have two sites for cloning. You can see here the linear DNA molecule with cohesive termini at both the ends. You have two positions of eco-R1 where you find the target site for eco-R1. In between there is a DNA segment measuring up to 20 kb. Now what exactly is done is this molecule is given with eco-R1 and given for digestion. So there are two cuts being produced and that 20 kb of non-essential region of the genome of phage DNA is removed. In that place you can add the DNA insert of your choice. So it goes without telling that you can insert DNA inserts of bigger size in replacement vectors when compared to insertion vectors. Okay. So up to 24 kb you can insert. Best example for replacement vector is lambda EMBL4. Lambda EMBL4, the bottom one. I have given the diagrammatic representation of how exactly insertion is done in my notes. Please refer to my notes. So EMBL4 can carry up to 20 kb insert DNA by replacing a segment flanked by pairs of eco-R1. Sometimes BAMH1 or SAL1, any one of these restriction enzymes can be used. Okay. Here in the diagram I have shown the eco-R1 sites. And selection and screening is not a problem at all. Recombinant selection with lambda EMBL4 can be done on the basis of size, or you can utilize the SPI phenotype. You can utilize the concept of SPI phenotype, SPI. I will tell you what it is. Now, so same thing, I have put it here, the details. Now, selection by using SPI. What exactly it is? See, phages cannot infect Escherichia coli cells which are already infected or which already possess an integrated form of a related phage called P2. Say if an Escherichia coli is infected by P2 phage, such E. coli cannot be infected by the lambda phage DNA and therefore they are said to be SPI. SPI means sensitive to P2 inhibition. SPI means sensitive to P2 inhibition. But some lambda cloning vectors are designed so that insertion of new DNA causes a change of the E. coli from SPI plus to SPI minus. SPI plus to SPI minus. See, to begin with, if the E. coli cell is pi plus, when you insert this vector with the DNA insert, the E. coli will become pi plus to pi minus. It will not be sensitive to P2 inhibition, and therefore in such cases selection of recombinant cells is very easy, because recombinant cells to infect that carry P2 prophages. Okay, so such change from pi plus to pi minus will enable recombinant cells to infect cells that carry P2 prophage. Okay, so such cells are used as host cells for cloning experiments with these vectors. Only recombinant cells are pi minus, whereas the others all non-recombinant cells will become pi plus, so that only recombinants form the plaques. So selection is by plaque formation. Okay. So this is about the replacement vectors, and best example is lambda EMBL4. What are the advantages of phage vectors over plasmid vectors? These are more efficient than plasmid vectors for cloning of DNA fragments. The largest insert size can be 24 kb in lambda phage vectors, while that for plasmid vectors it is less than 15 kb. This is one advantage. So insertion of large DNA inserts possible. Then it is easier to screen a large number of phage plaques. So in a culture plate than bacterial colonies for the identification of recombinant vectors. So two advantages are insertion of DNA of larger segment up to 24 kb instead of 15 kb. Then easy selection method. Okay. These are the two advantages of phage vectors when compared to plasmid vectors. Okay. Now, this is about phage vectors. Here I have given a table which tells you the differences or comparisons between insertion vectors and replacement vectors. Two key differences are: insertion vector is a type of phage vector in which a unique restriction site is introduced within the phage genome at the site of optional DNA. Okay. Whereas in replacement vector, here vector is developed from the removal of a middle filler figment region of the phage DNA. One difference. Second difference, the replacement vectors accommodate higher lengths of foreign DNA inserts when compared to insertion vectors. Okay, these are the two differences. Then, as per your syllabus, you have to learn one more type of vectors, that is cosmids. Cosmids, these are the hybrids between a phage DNA molecule and a bacterial plasmid. Cos is from phage part, MID is from plasmid. So, it is a hybrid of plasmid and a phage vector. Okay. So, hence the name cosmids. The design centers on the fact that, packaging of lambda DNA molecule into the phage protein coat need only the cause sites. Cause sites means cohesive termini in order to function. So what all is needed is just the cause sites from lambda phage. Okay. So that means the in vitro packaging reaction works like this. Not only for the phage genomes, but with any molecule that carries cause sites on either sides, separated by 37 to 52 kb of DNA. So, what is needed from phage DNA is just the cause sites. Okay, what exactly it is, I'll tell you a little later. Now, so you retain the cause sites and few other segments from lambda phage DNA, and in between you can add the DNA of your choice, and the formation of head or packaging takes place as usual. It is not hampered. It doesn't require only the genome. Instead of genome, any molecule that carries cause sites separated by 37 to 52 kb. The insert should not be more than this much. Okay. As usual, it is packed. Right. So, such a vector is known as cosmate. Now, what all the cosmates contain? Cosmates essentially contain 250 base pairs of lambda DNA. 250 base pairs of lambda DNA. What all it includes? One, I have been telling you the cos site, COS site, cos site or cohesive termini. What exactly it is? Okay, cohesive termini from 5' end to 3' end. It runs like this: G, 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 C, G, G, C, G, A, C, C, T. This on either side single-stranded molecule. Okay, single-stranded molecule. So, these cohesive termini, also referred to as cause sites, allow the DNA to be circularized after infection of the host cell, and cause site helps in circularization of DNA and plays an important role in packaging. So, this part of lambda phage DNA is retained. Okay. Then, second thing, sequences needed for binding and cleavage by terminase. Sequences needed for binding and cleavage by terminase, so that under appropriate conditions they are packed in vitro into empty phage particles. Alright. So, these are the two things that are needed from lambda phage DNA. Then rest you can take from plasmids. Okay. Now, a typical cosmate should have one ori of replication because autonomous replication is needed. Then it should have unique restriction sites. Then it should have selectable markers from plasmid. Selectable markers from plasmid and the cos site. Alright. So, these are the 4 requirements that a typical cosmate vector should have. Okay. Then, here I have shown you the structure of a plasmid. Cosmate. Structure of a cosmate. Right. So, this cosmate vector is known as PJB8. It has 5.4 kb length, and you can see here one ampicillin resistance gene, and most important is cos site, right? Cos site, and then there is one region for hint3 and sol1, and whatever is marked here with yellow, only that region has been taken from lambda phage DNA. Rest is from plasmid. Okay, there is one ori of replication, and there is one site for EcoR1. So, this is how a typical cosmate looks like. Alright. Now, so how exactly cloning is brought about in case of cosmates? Now, how cloning is done? The cosmate is opened at unique restriction site. The cosmate is opened at its unique restriction site, and a new DNA fragment is inserted. And then, as usual, ligation is carried out. Now, I have to tell you one more concept, what is known as concatemers. Concatemers. Okay, framing of concatemers. This is a new concept what you are going to learn with reference to cosmates. What exactly is done? See, you will cut open the cosmet at its unique restriction site and you will add the new DNA fragment that is to be inserted. Then ligation is carried out so as to frame the concatemers. What is this? During annealing and ligation of DNA insert, in fact, DNA insert is cut into many pieces. During annealing and ligation of DNA insert, two or more cosmets may join end to end. Two or more cosmets may join end to end, producing what is known as a concatemer, which is the proper precursor for packaging the genome into forge hosts. Okay. Now, if the inserted DNA is of right size, the in vitro packaging cleaves the cos sites and places the recombinant cosmets in mature phage particles. These phages are then used to infect an E. coli culture. Though the clear plaques are not formed, though we say selection is by clear plaque formation, clear plaques are not formed, then what should we do? Instead, infected cells are plated onto a selective medium and antibiotic resistant colonies are grown. You must be knowing here that there is one ampicillin resistance gene, right? So, that theoretically here one advantage is all colonies are recombinant. All colonies are recombinant because non-recombinant linear cosmets are too small to be packaged into heads. So by rule, if you get a colony, invariably it's going to be recombinant colony. Alright. What are the typical features of a cosmet? You can use, they are used to clone DNA inserts up to 45 kb. An advantage. Earlier, it was 25 kb with lambda phage. Earlier, 15 kb with plasmids, but here, the as big as 45 kb DNA inserts can be cloned. Then, packaged into lambda particles, which infect the host cells, it is many folds more efficient than plasmid transformation. Transformation is more efficient, okay, in case of cosmets. Then selection of recombinant vector is based on the procedure applicable to the plasmid which goes into the formation of cosmet, okay. What type of plasmid you select to design a cosmet will decide the type of selection of recombinant vector. Then these cosmets are amplified and maintained in the same manner as the contributing plasmids. So these are all the typical features of a cosmet. Dear students, this is all about the lambda phage vectors and cosmets. When you are back for offline classes, if further any details need to be given, will be given in the offline class. Thank you for your patient hearing. Thanks. Any questions, you do contact.